What's up, guys? Welcome back to the Strong.Sistas YouTube channel. In today's video, I had a great discussion with Kyle Mamounis all about PUFAs, polyunsaturated fatty acids. So Kyle received a PhD in nutrition science from Rutgers University, and he is now a postdoctoral researcher at University of Central Florida. So his graduate research was focused on different types of fatty acids, such as PUFAs, and their effect on disease. So in today's chat, we talk about PUFAs. What are PUFAs? What are potential consequences of having too many PUFAs inside of our bodies? And kind of the literature around PUFAs, how they've been prized, whereas saturated fats have kind of been demonized. So if you are interested in hearing more from Kyle, you can check out his research papers on his ResearchGate profile. You can also check out his YouTube channel at Nutrichronology. As always, this is not medical advice. Do your own research as to the best type of nutritional strategy for you. All right, so let's get into the PUFA discussion. All right, Kyle, thank you so much for hopping on today to talk all about PUFAs. But before we get into the discussion, can you explain to our audience who is Kyle Mamounis? Did I properly?
That's close enough.
Okay. Who is Kyle Mamounis PhD? What was your dissertation work and some of your main findings?
I did a PhD in nutritional science. I finished the work in 2016. My degree says 2017. It was at Rutgers, and I worked in an endocrinology lab, and the project that I did was the obesity neuroendocrinology, so mostly hypothalamic gene expression of different neuropeptides associated with like appetite and inflammation and things that tend to be dysregulated when people have things like diabetes or some type of I guess usually this would be like a clinical condition, but it's used as a model in animals for just obesity in general, like overeating.
Yeah.
And so I was in an endocrinology lab, but then I did a nutrition project.
Okay.
So I had some mice, and I basically did 3 chapters. One was feeding male mice 3 levels of, it was all the same percentage calories in the mouse chow that we bought from fat, but then the one was mostly saturated fat from coconut oil, and then there was a third one that was mostly, well, I guess like 60-70%, the 60% PUFA from things like safflower and soybean oil, the really highly unsaturated vegetable oils, and then the second one was in the middle.
Okay.
So I did that with male mice, then I did it with female mice, but part of that was because the lab I worked in was an estrogen lab, so we removed the ovaries from those female mice and gave half of them. So for that, it was 4 groups. It was saturated fat, no estrogen, saturated fat, replacement estrogen, on-site. So, and then the last group was an intergenerational experiment. So I bred mice and had their parents on whatever diet, and then weaned the pups onto high or low fat diets, and looked at the kind of, it was just a 1 generation effect.
Yeah.
And then, so that's what I did science wise. And then I've just always been interested in diets. That's why I decided to do a PhD. I've never really been interested in being a scientist for a career. I just did it because I wanted to, like, I was one of these people that was obsessed with reading and watching videos of, say like 15 years ago, like Gary Taubes or somebody like that.
Yeah.
And I just wanted to be part of that.
Okay.
And so this is where I ended up.
It's funny how kind of opposite side of the spectrum you kind of are now relative to.
Yeah. Well, if you go far enough into anything,
yeah,
like I don't know, like you want to be a musician, and you like some band when you're like 15, by the time you're 30, like if you're a professional musician, you probably are going to be playing a different style of music and maybe slightly embarrassed about the things you used to like. I don't know.
Yeah, I completely understand that. So what was your like main finding from your dissertation then?
My main finding was not in the hypothalamic stuff. So I was hoping to get a real nice, sexy data set of gene expression showing something like an upregulation of inflammation in one of the groups. And it was just kind of all over the place. The different markers, like I was looking at interleukins, and then, I was looking at like leptin receptor and all kinds of stuff that would be controlling metabolism and appetite centrally.
Yeah.
And there were a couple of differences in the groups, but there wasn't like a story really there that showed anything. But the one consistent effect was that glucose metabolism was disturbed more in the PUFA group, the highest PUFA group. So I think the males, it was different between the males and the females because I gave both of the mice at the end of the diet. I think I fed them for 3 months on these diets. And then we would do glucose tolerance test, an insulin tolerance test, body composition MRI. We put them in these boxes that would do indirect calorimetry and measure like their inhaled oxygen and exhaled CO2 for like a day. So these poor mice would just be in like a plastic box living.
Yeah.
Uh, and anyway, it was the glucose tolerance tests and the insulin tolerance tests that were different. So I think the male mice had really, really slowed glucose disposal in the highest vegetable oil group. And their insulin, the glucose disposal from insulin treatment, though, was, I think it was a little bit worse, but not quite as bad. And then it was the opposite for the females. The insulin didn't work as well, but the glucose disposal was kind of disturbed in both fat groups.
Interesting.
So that was the main finding.
Yeah. Well, that really aligns well with the teachings and principles of the repeat ideology. And so that's like a good segue into if we could just like briefly cover PUFAs, so different types of fatty acids and sources of each. So, I mean, obviously, we're going to basically have three types: the saturated, the mono, and the polyunsaturated, which is the PUFA. Can you briefly discuss those three?
Sure. So so all fatty acids, are made of two parts, what's called an acyl chain, which is just, carbon to carbon to carbon to carbon. And at the end of that, at the one end, it hooks up in what's called an ester linkage to glycerol. And there's a carboxy group at the end, and then that hooks up in an ester linkage to glycerol, and that's what makes a triglyceride or a diglyceride. So you got the carboxy end and the methyl end, which just ends in a regular old carbon. So it's just like a long chain. And if that chain is just carbon to carbon to carbon in single bonds the whole way, then what will fill up the rest of the bonds, because carbon always makes four, are hydrogens. And that's when it's called saturated, because it's just full of hydrogens. And if along that chain at any point there's one double bond, then the carbon will only have, the two carbons that are involved in that double bond will only have one hydrogen each. And that's called unsaturation. And so if there's just one, it's monounsaturated. And if there's any more than one, it's polyunsaturated. And usually the unsaturated bonds occur because they're created enzymatically by like bacteria and different, usually the like bacteria and lower order organisms are what actually produce these types of fatty acids. And like, for example, we can't produce PUFAs. We can only take PUFAs that we get, and we can extend them and desaturate them further. So like we could take an 18 carbon long PUFA, linoleic acid, and turn it into a 20 or a 22 or a 24 long carbon, and do that with not so much. Well, anyway, then there's the then there's the omega-6 and
Okay.
omega-3. That's the last distinction. So because mono.
So just real quick, so basically just like saturated chain, you have no double bonds.
Right.
And then unsaturated, you've got at least one double bond. So the mono group, that's going to be like the olive oil. What else is mono?
Well, olive oil has a lot of mono in it, but it also has some saturated fats and some polyunsaturated. But
Correct.
yeah, olive oil is mostly oleic acid. That's why it's called oleic.
Ah, okay. Well, so the mono has one, and then the polyunsaturated fatty acids have more than one. So there's just a different amount of double bonds throughout the chain. And so now you're going to get into, so if we're considering then polyunsaturated fatty acids, with more than two double bonds, we have the omega-3 and the omega-6.
Yeah. So the double bonds usually start at a characteristic. I mean, you could synthetically put the double bonds anywhere, but naturally occurring ones usually start at either what's called the N3 or omega-3 position or the 6. And that's counted from the methyl terminal. So it's like 3 carbons from the one all the way on the end, far from the glycerol, or 6.
Okay.
And like I said, we can extend and desaturate some PUFAs in certain ways with enzymes that we have, but we can't make prior double bonds. So if you consume an omega-6, you can't turn that into an omega-3 as a human, like with what we have. We can make a longer omega-6. And if we consume linolenic acid, which is the sort of normal 18 carbon plant omega-3 PUFA that you get in vegetable oils, or like flaxseed oil, people will take that specifically for that. We could turn that into a longer omega-3 PUFA, but we can't turn a 6 into a 3.
Okay. So within omega-3, we've got the like ALA, EPA, and DHA, right?
Mm-hmm.
And then for omega-6, we've got the linoleic acid, LA, and then arachidonic acid, ARA. Okay. And so I think you brought up a good point earlier. There's not a single fat source that we consume that's just like 100% any of these, right? Like that's natural in like a diet form, not talking about supplements. All of the fat that we consume is some combination of these. But like, for example, like beef fat is considered a saturated fat because it's significantly higher in saturated fats than the other ones.
Yeah.
Okay. I think that
but sometimes that's only like I there was a time when I had all of this memorized off the top of my head, and that time is over now. But like just like kind of a ballpark estimate, beef fat might only be 60% saturated, or even 50%, and then maybe like 20% monounsaturated and like 30% or less polyunsaturated. So people will call that a saturated fat, but it's not like it's 90% saturated.
Yeah.
Now, butter is much more saturated than like beef carcass fat. And then coconut oil is, I believe, one of, if not the most saturated natural sources. It's like in the 90%.
Yeah. So in general, the fat sources that are higher in saturated fats, such as coconut oil, butter, those are solid at room temperature. And then the fats that are higher in the PUFAs are going to be liquid at room temperature. That's
yes.
like the general rule of thumb.
Mm-hmm.
Okay. So what I find really interesting about your work is like, clearly you were really in depth in the fatty acid discussion for your dissertation, and writing a dissertation is no joke. You have to have a really good handle on the literature. Most dissertations have some sort of literature review. So I've heard previous talks where you've talked extensively about kind of the research around PUFAs and saturated fat. And it's interesting, Ray Peat kind of talks about how he went to school a lot longer ago, and the picture that the research painted about fatty acids and PUFAs and saturated fat was quite different than the picture that the literature paints now. Is that like, so basically like early 1900s versus like the 2020s now, where kind of saturated fat is now demonized and PUFAs are almost praised. Why has saturated fat been demonized? Like, where did that kind of come from or stem from?
As far as I know, it's the normal heart health story, which is that heart disease became a target of, treatment in the early 20th century, and then sort of ramped up even more in the kind of World War II, post World War II time period. And one of the early factors in that was cholesterol,
Okay.
being found in atherosclerotic plaques, which were, found in people that were, had like died of heart disease, for example, or a heart attack or a stroke. And then they tried to reproduce these things in animals, and they found that consuming cholesterol can sometimes cause the blood to have more cholesterol in it, but the types of fats you consume were even more of a deciding factor. So if you consume a diet that has a lot of saturated fat in it, you will have more serum cholesterol, like everything else kept equal, than if you eat that same diet but just substitute unsaturated fats, especially polyunsaturated.
So
And
it
so since cholesterol in the blood was considered bad, then saturated fat was considered bad.
Okay, so it purely comes from the whole like lipid hypothesis where high levels of cholesterol is bad and consuming saturated fat raises that, whereas consuming PUFAs, polyunsaturated fats, reduces your blood cholesterol levels, therefore it's good. So that it kind of purely comes from that.
Yeah, I think there's also, or maybe one of the things that made this take off even more than it might have otherwise, is there's always kind of been a skepticism, like in the culture for many, many centuries, about meat eating. And there's kind of, that like eating animal fat is like gluttonous, and that, like, for example, most fasting rituals, if they don't include not eating anything, like one of the first things you cut out in any kind of like spiritual or health fast would be like animal foods,
Yeah. Yeah.
So there's kind of always been this idea that like really heavy, rich, fatty animal foods are kind of suspicious. And I think that plays a part in this. It just makes sense to people. Like even that really stupid stuff, like you'll see on TV where somebody will just have like a beaker of cholesterol and show that it's like thick
Yeah.
and say, oh, you don't want this in your
Yeah.
arteries,
Yeah. And can you briefly touch on that? That seems to just still our dad had a stroke about a month ago now. And it
Oh,
was incredibly frustrating to read through the documents that he received from the hospital coming home and like what the suggested diet was. Um, and one of the things was like avoid saturated fat and use vegetable oils for cooking and like that kind of thing. And like I, of course, kind of have like a freak out because I do not agree with any of those things. But then I'm going to pull up something really quickly. Like my dad will then search online about these things, and you go to like Harvard. How can you deny Harvard, right?
right.
So
Well,
swapping
it's getting easier every day.
that's true. That's true. But swapping saturated fat and carbs for linoleic acid, the main PUFA found in vegetable oil, nuts, and seeds, lowers the risk of coronary heart disease, according to a new study. And like, it's just crazy to me that this information is still out there. And it was, I think, became incredibly frustrating when I saw that it was going to be applying to like my dad too, where I just it seems to just still go back to the theory that, all right, if you lower your saturated fat, your cholesterol is going to go down, therefore you're going to have less clotting and you'll have your risk for another stroke will go down.
Mm-hmm.
So can you briefly touch on like how did PUFAs become so praised? Like where did this kind of stem from, and how has it kind of taken over mainstream health recommendations?
I think there were two main inflection points. The first one was when it was discovered, in the early days of the lipid heart hypothesis type study, where it was found to, just lower serum cholesterol. So
Yeah.
that was like the first thing. And then the second leg was the omega-3 stuff that took off. I forget when like the seminal studies were done, like the 70s, 80s. It really took off in the 90s in the scientific literature. where there were a lot of big studies done with omega-3 supplementation, like fish oil and things like that, and endpoints like heart disease, and then also biomarkers for heart disease, which is another thing to look out for. Whenever you're looking at a study for something like heart disease, you want to make sure that the endpoints they're talking about aren't just assumed. Like, for example, there are papers that will say this thing lowers heart disease or heart disease risk, but what they're measuring is serum cholesterol. But then that kind of just is begging the question, like, if it's not a study that actually measures like a group of people and how many have a heart attack, then you have to just make sure, that you're not getting sort of like well, that's the question I'm asking, right? Is that, are blood lipids bad? Like, you can't have presence of blood lipids as the bad thing. But yeah, and then in the popular culture and marketing and stuff, I think it was, the 21st century when the omega-3s started to take off from the previous decades of, it kind of trickled in through products, and people started taking fish oil at first it was for the heart disease, benefits. And then people started noticing, especially older people who had problems with arthritis, they would get some relief from, like joint pain and things like that. So I think a lot of people took it for that, sort of like a really low, less strong cortisol almost, like if you have like a pain here or there.
Yeah.
So I think those are the two main things that like thrust it into popular consciousness. And there wasn't a lot of opposition because people are always suspicious of animal foods, like even just looking at animals, like you look at a cow, you look at a pig, they're fat. Like it's just, you
Yeah.
usually think, oh, it makes you fat, and fat is like a heart attack. Fat is, it's all kind of like these associations that you see. And then you have a doctor telling you that, and it makes sense to people.
Yeah. Okay. So I think so you're bringing up the fish oil. I think that's a good point to go down, because that is definitely controversial. I don't know of many people who will argue against someone in like the alternative health, versus like mainstream, who will argue that vegetable oil is a good thing, right?
Mm-hmm.
Because linoleic acid, omega-6s, they're bad. But now you see a lot of people pushing like high omega-3, for example, fish oil. And like you said, it's like heart healthy. And then some of the other things are like, it's like blood thinning, anti-inflammatory. I just have some notes written down from a few papers that I pulled up. Anti-inflammatory, it produces resolvins. It changes the fluidity. It improves your cellular metabolism, and there's HRV improvements. So it just seems like there are a group of studies that clearly document, or maybe just their title is good at summarizing, that they document these benefits of fish oil supplements. You still see it readily used in like the bodybuilding community, the alternative health, the mainstream health. So I know that you have some conflicting views on omega-3s and fish oil pills. If you want to dive into that a little bit about why you think there may not be as many benefits as people say, or how, then how do you explain the benefits that are shown in these studies?
Well, yeah, there's a couple different like sub discussions within there, because the omega-3 story, it's really big.
Okay.
The first one is that the original studies, and I should have just kind of brought them up, like the historical, omega-3 literature, but anybody that's into this stuff knows what I'm talking about. It was mostly heart disease trials, and there were some pretty good results in the second half of the 20th century.
Okay.
And then towards the very, very end of the 20th century, but really into the 21st century, so the aughts and the 2010s, some of those were attempted to be replicated, and the replication was a huge failure, which is why people don't really like, they talk about omega-3s being heart healthy, but it's really not pushed. I mean, they will say consuming unsaturated fats in general, like the American Heart Association or something has like the official line that, replacing saturated fats with polyunsaturated fats is heart healthy.
Yeah.
But there's really not good evidence anymore that taking supplementary omega-3 PUFAs, whether it's like fish oil or synthetic or whatever, does that, specifically improves heart health. So they moved on to these other things. And then you just get into this really, because what's going on with fatty acids is super complicated. And there's a few times I've touched on this, but like you can find papers that will say a protein like interleukin 10. There are papers where that will be an endpoint that it like increases in an animal or sometimes in people, but usually this stuff is done in animals because, you can take a lot of their blood, you can kill them and, look in their tissues and stuff. That'll be an endpoint for inflammation. And then in other studies, that'll be an endpoint for anti-inflammation. And then you'll have ones that will say, well, it induces inflammation and resolves inflammation. And all of the different proteins and biomolecules that are associated with inflammation, which is a pretty hazy concept to begin with, could be said to be inflammatory because they're associated with inflammation. And some of them people don't really call anti-inflammatory, but many of them are also called anti-inflammatory because they're involved with starting inflammatory processes and resolving them and ending them. So it's very, you really have to, that's one of the parts of the literature that it's not straightforward. It's not like, okay, this group has higher fasting blood glucose, so they have like symptoms of diabetes. Like it's not a this thing is present, therefore this thing. It's like if you saw a process where somebody drops something off, somebody picks it up and takes it somewhere else, and somebody takes it to a third place. Like if you didn't understand that whole process and you just saw one of those steps, you would not understand what's happening.
Okay.
So with things that are produced, things that are upregulated when you have more omega-3 fatty acids, they can be interpreted actually as inflammatory or anti-inflammatory. And the same thing with omega-6, and those two things compete with each other. So if there's a bunch of eicosanoids, which are the large family of metabolites from the polyunsaturated fatty acids that get made, things like prostaglandins. If something is inducing inflammation, even something like a cut and it gets infected, or something more metabolic, more chronic, you'll have those things being made locally. And if there's a bunch of omega-3 present, it'll compete with omega-6 for access to the enzymes that make those eicosanoids, and it'll change the molecular signature of the inflammation. And that can be interpreted, unless you actually have a really nice endpoint for like what's going on in like the tissue or whatever you're studying. Some of these molecular things where people just decide, okay, we're looking at these 4 interleukins, a couple of cyclooxygenases, like chemokines, whatever, and we're just going to say that these things being present is inflammation. That could be true, or it could be like resolving inflammation from something. So that's where things get kind of a little, like the literature can get really crazy because some of the people doing these studies don't have a broad view of physiology like that. They just, they'll start doing experiments in a lab, and that lab studies, like one of my advisors, her lab was exercise. She was in exercise science, and she also looked at gut health and so exercise, the effects of the microbiome and gut health, and omega-3 supplementation. So she would like give mice diets that had, supplementary omega-3s and look at their microbiome, and then have some of those mice do exercise and see if that, and it's this whole thing. And if you just think of it as, oh, look, the gut microbiome is more diverse. And do you know what I'm saying? Like, it's kind of hard to say sometimes.
Yeah.
It's not like a heart attack. It's not like you've got 10,000 people here, 10,000 people there. These people are all given fish oil. These people aren't.
Yeah.
And the people that are given fish oil like don't have heart attacks. It's not like that. And the studies that were like that have utterly failed in replication. There's just like a tremendous, it's basically, they're not even trying anymore to have like large clinical trials of omega-3 supplementation for heart disease, outcomes.
So basically, is this kind of a accurate summary? So a lot of the papers that show benefits of omega-3 on like a specific thing, such as like reducing inflammation or something, that's kind of targeting a specific molecular endpoint that can be interpreted in a few different ways. It's kind of like almost too dialed in. And then if we look at, like, for example, control trials with humans, the same things don't necessarily hold up with, for example, like heart disease.
Well, with heart disease, they don't. With inflammation, I'm not sure. I'm sure that there are probably studies because, like I said, they compete with omega-6.
Yeah.
And there's one of the things that I try to emphasize is there's a pretty big difference between acute and chronic effects with fatty acids, specifically with PUFA, because we're always going to have saturated fat in our system because we make saturated fats.
Yeah.
So
I learned this from either you or discussion with Danny that our body, when it makes fatty acids, it will preferentially make saturated and mono. It will not make PUFAs. So that's kind of telling there a little bit.
Yes. Yeah. And actually I just was thinking of, I don't know if this is a novel theory. I was going to write something up about it, but if you think of it that way, right? So there's always going to be saturated fat present in a system, in a human system. If you're eating a super high carbohydrate diet, like some island population that eats just like starch and, a little bit of fish or something, they will have their fat will be very saturated and a little monounsaturated because they're producing most of their fatty acids de novo. And whereas if you are consuming fat dietarily, you're going to have more PUFA coming in than if you're not consuming fat. In other words, the presence of dietary PUFA is a signal to your body that there is fat present in the environment. Whereas the presence of saturated fats is not necessarily a signal of that. Although, I guess if you're consuming a lot of saturated fats, your body would get the message. Like if somebody's on a low carb and a high fat diet, their body will get the message that, do more lipolysis and burn more fatty acids for energy because those are present. But if you're eating a mixed diet with sufficient carbs that you're not getting into a lot of lipolysis or something like ketosis, then dietary PUFA is sort of just a signal of dietary fat in general. Because even like we said, even if you're eating a land animal that is mostly saturated, you're getting a decent amount, a lot compared to what you're producing de novo, which is basically nothing, of PUFA. And a lot of the things that PUFA do that people have demonstrated mechanistically is, there's these receptors that pick up fatty acids, the PPAR receptors, peroxisome proliferator activated receptors, and they react to the presence of fatty acids and cause more genes to be expressed involved with fatty acid oxidation, which makes sense. So it's like fatty acids are present. And PUFAs are more, so there's PPAR alpha, gamma, I think delta. I know gamma is the one that is more specific to PUFAs, and the other two are a little bit more promiscuous, but PUFAs are better at binding them in general. Okay. And so this is consistent with if there's fat in the environment, animals or even plant fat that you can get, you're going to have more PUFA than if there's very little fat available in the environment. It activates these fatty acid oxidizing, these receptors that like upregulate the whole thing of use fats and also make less endogenous fat. So it could just be that this entire thing where PUFAs make you produce less fat de novo, export less cholesterol from the liver, is just because it's telling your body like, oh, we're getting lipids from the environment. Because if you're getting natural lipids from the environment, you're also getting cholesterol, right? Unless there's very little human populations historically that would get all of their fat from a plant source.
Right.
So you would be getting cholesterol, you would be getting saturated, monounsaturated, and polyunsaturated fats. And that would tell your body, okay, oxidize more fats than when you're not getting these inputs, and you can produce less fat, and you can produce less endogenous cholesterol as well. So those endpoints, like we were saying, for reducing circulating cholesterol, are seen as something that's like a benefit, but it really depends. Like that doesn't necessarily mean that there's less like atherosclerosis building up just because there's less cholesterol at that time or in a short-term effect from taking something. And it's basically the situation now where we have access to fat all the time is pretty novel. So, not to get too like paleo, about the whole thing, but in the past, I think a lot of these physiological effects that our bodies have to the presence or absence of certain nutrients, or like the ratios of whatever, were a lot more functional. Like, oh, okay, it's this time of the year, so there's access to like, fructose, to certain types of fats, to this or that, and it upregulates these processes. But now we basically have access to everything all the time. So
It all.
yeah, and if people don't think of it that way, and they don't kind of think of like, where have these processes come from, because that framework, not that you have to think about it all the time, but that can help you understand why, oh, these genes are being upregulated from the presence of this thing.
Yeah.
And if you don't have a framework for thinking why that's happening, you could be like, oh, this is good or this is bad. But it might change your perspective on whether something is good or bad, or maybe good sometimes, but not all the time, or whatever. If you think about, why something would be happening.
Yeah. Yeah. Okay. So back to the omega-3s really quickly. So I did a little bit of like looking into the literature, and it's just like, it's really hard to deny the benefits that are documented about like brain health with like DHA. So
Mhm.
again, omega-3, there's the ALA, EPA, and DHA. And there just seems to be like so many studies documenting about how like with low DHA, you're going to see brain degeneration. And for example, there's lower risk of preterm birth with DHA level above 5%. The brain and the retina themselves are rich in DHA. So I think that you would only get a lot of DHA levels based on what we were just talking about. You'd only consume high DHA if you're consuming the PUFAs, because our bodies aren't necessarily going to make those themselves. So what are your thoughts on the DHA side of things and like the brain health?
That's probably the area that I've looked at the least, but I'm not like Ray Pete level anti-PUFA. And like, for example, on the question of essential fatty acids,
Yeah.
I'm, I guess, agnostic. I'm not 100% sure, because it's very difficult to, but also essential is just, like glycine, for example, is not essential, but a lot of people think dietary, it's essential for a certain level of health, outcomes maybe. So it's very hard to produce so-called essential fatty acid deficiency in humans, because we live a lot longer than like mice. So to actually induce a deficiency that maybe you could induce it in a mouse or a rat in like a few months of a fat-free diet or something like that, and start to see symptoms, that might take years for a human. I mean, nobody's going to do that. So however, I don't think that, when these fatty acids, in addition to being oxidized for fuel, they end up everywhere. They end up in phospholipids, in cell membranes, in the membranes and structures within cells, all over the place. Like you said, the brain, they're put into mitochondria. I think there probably is an appropriate level of, diverse fatty acids integrated into cellular constituents. And I think that that level is probably pretty close to, if we could kind of figure out what the balance input output would be from like maybe an historic human diet. So we know that we don't produce them. So if they're essential, that means that we were, eating them maybe intermittently.
Yeah.
And, people have been eating fish, and, it's for a lot, like, any civilization that's like by a body of water, fish are considered a good food.
Right.
So to me, it makes sense. Also, like, even omega-6, so linoleic acid gets metabolized to arachidonic acid. And that's the more bioactive in humans. It's the one that gets made into eicosanoids, and it also gets made into endogenous cannabinoids and all kinds of metabolites. And it has this preferential, it gets stuck in the 2 position in triglycerides and phospholipids in general. And there's enzymes that do things with it that are so specific that it seems like, yeah, it's meant to be there. Like, we're meant to have this thing at some level.
Yeah.
It's just a question of, like, is it good to have, like, how modern Western, like, I guess the Anglosphere, I don't know about the continental Europe, maybe 12% of their calories come from vegetable oil, fatty acids. And then if they're taking fish oils on top of that, however much is PUFA in total. That, I think, especially with the vegetable oil, is much more of like a simple, okay, this is just a bad idea kind of thing.
Yeah.
But I wouldn't really advocate that people don't eat any fish, or don't eat any sources of, I think we probably need omega-3 at some level, and, it's present in meat anyway.
Yeah.
So, and if you keep the omega-6 down,
Yeah.
you will have more tissue omega-3. So a lot of these endpoints, right, like integration of some of the longer chain omega-3 fatty acids into these different tissues, that is driven as much or more by the intake of omega-3 as by the non-intake of omega-6, because they compete for the same enzymes to get moved around, to get modified, to get stuck into triglycerides and different phospholipid products. So, like, one of the best omega-3 supplements you can take is to stop consuming like vegetable oil, and I think that's a safer way to supplement your omega-3.
That's kind of the approach that my sister and I kind of take now, learning about some of the potential downsides of excessive PUFA intake, but recognizing that you can't, if you're going to consume fat, you can't avoid PUFA entirely. And we are consuming a lot of like ruminant meat with fat on it. And so going to get some amount of omega-3 in there. We'll have fish on occasion. Granted, we don't really prioritize like super fatty fish, but like we'll have like a lot of oysters and crab and shrimp and other types of like the leaner fish. And then if we feel like salmon, we'll have it. But I think that this is the challenge in like the nutrition space. As soon as one sees a headline of this is good, then it's just this like more is better. Can you possibly touch on some of the potential downsides of too much PUFA consumption? So again, to remind the audience, like PUFA is polyunsaturated fatty acids, omega-3 and omega-6. And so technically foods that are higher in omega-3 are still like a PUFA. But I think there's a huge difference between like whole food form of PUFA, which contains like, for example, vitamin E and things like that, than these obviously vegetable oils that are reduced down to like a single nutrient and don't really contain many other beneficial compounds. So can you talk about some of the potential downsides of excessive PUFA intake?
Yeah. One of the things that I'll mention first is the vitamin E connection. I've been trying to find, so one of the first things that people will say is, even if PUFA, oxidize, and that would be one of the really simple ways that it could be bad in your body, is that it can be just oxidized, in tissues in circulation, non-enzymatically. So just kind of by accident, and produce your reactive species that then go on to can oxidize other things, other fatty acids, and then other cellular constituents, consuming eventually, consuming antioxidants that could be doing something else. But vitamin E, is usually, as you said, prepackaged with most natural sources of PUFAs, which
Yeah.
makes sense because the organism that's producing them, whether it's a plant or an animal, is also not interested in excessive, auto oxidation of its fatty acids, which would be
Right.
dangerous for anything. So, however, people and I have yet to be able to find good sort of kinetic data on this, and eventually when I do, I'll try to write something on it. But when you take in fatty acids, they go all over the place, right? Some of them will get burned for energy, some of them will be stored, some of them will end up like just in fat cells themselves, but also like any kind of cell. They could just become a phospholipid and become like a skin cell membrane constituent. They can go all over the place. And the vitamin E that you take in with them will move throughout tissues as well and in circulation, but it doesn't necessarily, it's not like every PUFA or every 10 PUFAs has a chaperone vitamin E or two that it takes with it. Right.
Yeah.
And even if it did, PUFA, if there's a PUFA floating around, it encounters, say, unliganded iron and oxygen, or even worse, a reactive oxidative species, and it becomes oxidized. But there is vitamin E around, and it reduces it back. That can happen a certain number of times, but the vitamin E eventually gets consumed. But the PUFA does not get consumed. So eventually, over time, unless you're burning, like in some way disposing of the PUFA and not storing it at the same rate that the vitamin E that came in with that PUFA is being used and metabolized and no longer present, you will have a buildup of potentially oxidizable fatty acids, whether it's in your fat stores, which would then be liberated with lipolysis during fasting or like low carb diet, or in other tissues, or like all the way into your mitochondria, which is a very oxidative environment. It's like a crazy engine. At some point, there will be an imbalance. So if you had a study, and this is one of the problems that I see with a lot of the studies, if you give somebody a bunch of like sunflower oil or any of these things, it has a lot of vitamin E in it. And if you look at those people for 3 months, they may have less oxidized fats that you can observe in their blood, in different tissues, than somebody who is not consuming that sunflower oil, because the vitamin E is the primary player over that time period.
Okay.
But if you keep consuming those fatty acids, you'll store some of them, and the vitamin E will not stick. It essentially will have a shorter half-life in your system than part of the fatty acids. Like if you consume a gram, I'm not going to be able to give numbers here because I don't know. I can't think off the top of my head, like if you have so much sunflower oil, how much vitamin E is in that. I would have to look that up. But whatever's prepackaged together, unless you have almost no fat stores, like you're one of those like people that can't store, like have those fatty acid storage diseases.
Yeah.
You're going to have fatty acids have one of the longest half-lives of anything that you can take in.
Yeah.
The ones that get stored. So like I said, some of it is going to get put over here, used for this, that, get excreted, get turned into metabolites, and some of it's going to get stored. And all of those different things have different lengths of time that you can expect it to be present. So over a lifetime of consuming a lot of polyunsaturated fatty acids, you will have a buildup of those in storage, in tissues, in other places. You won't have a buildup of vitamin E. There's only so much you can take in, and it turns over more. Because that vitamin E, it's not there to protect the PUFA that it came with. It's promiscuous. It might get pulled off to reduce an oxidized species from something else,
Yeah.
like you could be a smoker and take in some reactive oxidative species from that's in your system, and the vitamin E gets consumed from that. Oh, well, now it's not there for the PUFA that you took in. And obviously there's a million things that could happen, right? So most of the human studies, just by the nature of it, they're super expensive. It would be almost impossible to follow people and give some people like a cup of sunflower oil or something to drink every few days, and another group not, for like 20 years.
Yeah.
But what I think is, if you do that, right, so on a population level, we have replaced cooking oils that, used to be more like animal fats and maybe a little bit more olive oil, things like that, with these vegetable oils. I mean, if you just walk into a supermarket, you can see aisles of these things in plastic containers, clear plastic containers at room temperature with like full, lights on them, which will not cause a lot of oxidation quickly, but there will be some.
Yeah.
Those have, taken over as the primary cooking oils, and also in things like salad dressings. If you walk down the aisle of salad dressings, just normal,
Yeah.
not like health food stuff, it's all soybean oil, maybe safflower, canola, things like that. Most of it's soybean oil. So now you have this basically replacement of quite a bit of fat intake from, what would have been much more saturated to polyunsaturated. And over time, chronically, that ends up in your LDL cholesterol, and it can be oxidized there, which is a major way that forms foam cells that create atherosclerotic plaques. It ends up in mitochondria. That's one of the things I was just looking up, is the literature is you can see, it's funny if you look at the years of these things. It used to be, like, in the 20th century, pretty understood that when mitochondria took up a certain amount of PUFA, including omega-3, like either omega-6 or omega-3, they would get more fluid, but they would also start to consume more of the oxygen that they were pulling in for respiration to oxidize themselves. And sometimes it would just straight up kill cells and cause apoptosis in like over unsaturated cells with really, really unsaturated mitochondria, or it just, becomes more and more of an oxidative burden, and they kind of stop working. And then you can see there's some literature as you get closer to the end of the 20th century, when omega-3 started to become a popular product, where they were showing some benefits of mitochondrial uptake and, association into the membranes of omega-3. And it's really hard, because when you're doing something like what I do, you have to read the whole study, because sometimes, the title will say something, and then you read the study and you're like, wow, they're really being generous, or it, like they really picked the one thing good to
Yeah.
say about, and then, but even now, like even in the 21st century, you can see like all like okay, for example, just real quick, I have a few that I pulled up, and from the titles: influence of mitochondrial membrane fatty acid composition on proton leak and hydrogen peroxide production. So you can guess, like, fish oil and corn oil increased hydrogen peroxide production in liver mitochondria. Fish oil increases mitochondrial unsaturation, upregulating reactive oxidative species and apoptosis in rat colon cells, colonocytes. So those sound bad, but the same endpoints, and I want to emphasize this, you can dress this up. So there's another study that finds the same thing, but upregulation of mitochondrial antioxidant superoxide dismutase underpins persistent cardiac nutritional preconditioning by long chain N3 PUFAs in the rat. So those might sound, I mean, of course, that's like word salad to somebody that's not understanding all those words. But basically, the one thing said, some of the titles I read said, hey, hydrogen peroxide is getting produced. And this is causing an upregulation in enzymes that take care of that hydrogen peroxide. This one just says, hey, look, it's an upregulation of enzymes that are antioxidant. Like, this is great. But that's like saying, like, say crime increases in your town or something, and then there's more police.
Yeah.
Like, and then you say, hey, there's more police, so this is great. Like,
Yeah.
and but they're the same. Obviously, if there's a bunch of like hydrogen peroxide, which is as close to a universal toxin. I mean, there are hydrogen peroxide, like if there's a biochemical reaction, often has to be produced in a transition to make a product, right? But when it gets out into the general system, you just don't want that. Nobody wants it. Bacteria don't want it, animals don't want it. They use it to kill each other, and so when you see that getting made, it's a pretty big red flag when that's leaking out of mitochondria, or it's, getting the presence of it is increasing in the mitochondria. And so of course the cell is going to, if it can, produce something to contain it and to de-radicalize it, to reduce it back down to oxygen and water. And you could call that an increase in antioxidant production and say it's good. And that's what a lot of these, and perhaps not all of them. I mean, I'm sure there's some studies that I could read and they have a reasonable endpoint assay and they show some benefits of this or that or the other thing.
Yeah.
But a lot of them are kind of silly like that. And like I'll get in arguments with people online, and they'll say, oh, and they're just, because there's like a million studies. You can just rapid fire.
Yeah.
Nobody could read all the literature of omega-3. And that's just one subfield of like, diet physiology or whatever. So there's always a study, just about always, that could show you could show hydrogen peroxide is good.
Yeah.
You could show anything. There's probably a study that's like mercury is good or something. So you have to understand, like, you have to step back and you actually have to have a worldview. You have to have like, what is the organism trying to do? Like what's sort of a normal operation and what's like, for example, if you understand that police activity would go up from crime, right? Then you can understand that increased police activity isn't necessarily a good sign.
Yeah.
Like, but if there was like an alien that didn't understand that and they just knew that like police stop crime or something, they would go, okay, great. Like this must be a safe neighborhood because there's cops everywhere.
Yeah.
But we could understand that the opposite.
Yeah.
And it's the same with health. Like you have to have like a forensic way to look at these things that, understands the broader context of like, A to B to C.
And the alien view is very easy to just extract these like buzzword taglines and headlines that can then just be blasted and people only read those. And it seems like that's definitely been done these last couple of years. But okay, a lot to unpack there. One, I just want to go back to this really quickly. So you mentioned, I think most people know this, but just want to clarify. So this idea of oxidation. So unsaturated PUFAs can oxidize because of this double bond, right?
Yeah, it's basically, anything can be oxidized, right? And like anything can either have electrons taken away from it or be given in oxygen, which is like just chemical oxidation.
Yeah.
But different, molecules are more stable in their current configuration and the activation energy would be higher. So like you could oxidize a saturated fat if you blasted it with, say, a bunch of oxygen on a metal, like something that has a bunch of metal ions as catalysts with a high heat, right?
Yeah.
And then when you get to monounsaturated, that too is probably something that doesn't happen very much in the system because just the one double bond, it still requires. I tried to find activation energies years ago of these reactions, and it's kind of funny. Like some of that basic chemistry is almost lost to time because people don't know it anymore. I'm sure there at some point somebody did something like that, like an experiment to see how much energy it takes to insert an oxygen into this type of acyl chain and this type of acyl chain. But when you have a polyunsaturated, where there's a double bond and then a single bond and then a double bond, the carbons in the middle of those two double bonds, they're flanked, are very good targets for attacks by reactive oxidative species, for things that want to take their electrons away, because they're surrounded by these double bonds, which have a more omnipresent and shared electron cloud than single bonds. So they have a more stable configuration if they lose an electron than something that is just single bond, single bond, single bond, single bond. And that's usually how reactions are determined. So the activation energy is proportional to the stability of the product. So if a product is, you could consider it like, if this product is unlikely to exist, or it's difficult for it, it's unstable, then it won't get made, because it will cost a lot of energy to shove everything into that direction.
Yeah. So,
And the more unsaturated bonds, the easier it is to shove some of those carbons into the direction of getting an oxygen attached.
okay. So you're saying, so this is an example of a PUFA, linoleic acid. So it's these two double bonds. So these are more readily oxidized than relative to a saturated fatty acid. So what are the conditions that are required, like the environmental conditions around this fatty acid that are required for oxidation?
Well, there has to be oxygen.
Yeah.
And it helps if it's, so what really helps if it's something like hydrogen peroxide or an oxygen radical. Molecular oxygen can do it, but it's just less likely. So all of these things are kind of in the sphere of probability.
Yeah.
So, I mean, actually, it could happen that a saturated fat could be oxidized in the system. It's just so improbable
Yeah.
that it's like not relevant, right? So it usually requires molecular oxygen, or even better, a more reactive oxidative species,
Or like,
and a metal catalyst ion. I think most people consider iron,
iron.
unliganded iron, as like the main culprit for us, just because that's something that can happen more often. But I'm pretty sure if you had copper that was just floating around and didn't have any chaperone proteins or wasn't stuck to anything, and perhaps some other metal ions, I'd have to relook at like which ones have the same kind of electrical properties as iron. But yeah, basically a catalyst,
Okay.
because that's how you do chemistry. So if you don't have an enzyme,
Yeah.
which is what's so nice about being a living organism with enzymes, is we can do a lot of reactions that cost a lot of energy synthetically, to make, to oxidize fats costs a lot of energy. You have to, blast it with heat and have some sort of catalyst, whereas we can just oxidize fats with our enzymes in the mitochondria with a very little energetic cost, because the cost was kind of paid up front by producing those enzymes in the first place. And now we have this like factory, that's like
Yeah.
really efficient at doing it.
Yeah. Okay. So iron, oxygen, and heat are three things that can increase the probability that a PUFA will be oxidized. Is that right?
Right. And so, of course, our bodies are pretty stable at the temperature that they're at.
Yeah.
But that gets back to the question of like, well, why is it that warm-blooded animals such as ourselves preferentially de novo lipogenize, the lipogenesis, make saturated fats, right? That question starts to answer itself when you think about what temperature does to increase the likelihood of autooxidation of fatty acids. And then, of course, plants like coconuts, which are tropical, are highly saturated. And then, seeds from more northern climate, or I guess, cold, either north or south
Yeah.
direction, are more unsaturated. And then cold water fish are more unsaturated than warm water fish, and et cetera, et cetera. And that is a extremely consistent theme across all of Earth's nature.
Yeah.
And so, that's obviously a really big piece of like evidence that that this is something that, at least as an organism, you have to worry about.
Yeah. Like
If you have a high temperature.
if salmon in cold water fish were high saturated, they would be stuck and they wouldn't be able to move.
Right. Yeah, they would probably have some sort of rigidity issues.
Yeah.
And it'd be interesting to see like what would happen if you took a salmon. Now, I don't know what their like an actual individual salmon, what its ability to change its own fatty acid profile is over its lifetime. Like if you grew salmon in warm water, would they themselves start to change, or is it they have like probably a locked in percentage? But basically it probably wouldn't be very healthy to, if you wanted to like farm salmon, to farm them in like bathtub water, you
Right.
know?
Ray Peat did talk about some sort of study where they were able to change the fatty acid composition of salmon based on the feed that they gave them.
Mm.
But again, not the environment, more just like the food that they gave them. But they've got to have some spectrum or else they can't function properly, I'm sure.
Oh, that's true. Because fish, since they're also animals, they get most of their fats from, it's really like microorganisms that sort of decide what fatty acids are, because like for example with cows, they produce some fatty acids, and then what they get that's not saturated is sort of decided by the bacteria in their ruminant digestive system. Like that they'll saturate some of the polyunsaturated fatty acids from like grass or grain or whatever the ruminant animal's eating. And that's why you'll get some of those naturally occurring trans fatty acids like vicinic, I think, because there's like one last step that they didn't do before it just ended up being absorbed by the animal in changing it from an unsaturated to a saturated bond. So like fish, cold water fish, they're consuming like smaller fish, and then eventually it gets down to like microbes in the water that are being eaten, up the food chain. And those are kind of deciding the fatty acid profile of the environment.
Yeah. Yeah. Okay. So then you also brought up that fatty acids have a long half-life. So what we consume, the fat that we consume helps build certain things in our body. Like you said, it gets kind of shuttled everywhere, and fat can stay in our body for like 2 to 4 years. Is that correct?
Yeah, I've seen those numbers, put around. I'm sure like it's probably radically different if you have a big turnover, if you like exercise a lot, or if you're somebody with a high metabolism, versus somebody else. There's probably a pretty big range there. But yeah, I think on the order of years is what you'd expect for somebody with a normal amount of fat stores to have like close to a 50 to 100% turnover.
Okay. And then alongside that, you also mentioned like, I kind of imagine in my head that PUFA and vitamin E, when consumed in like real whole form, they would like travel together. But you bring up a great point that it's kind of just, again, it goes back to that probability of oxidation. Your PUFA gets put somewhere in your body, and when it's faced with, for example, iron and oxygen, whether or not it oxidizes and causes these downstream effects depends on whether or not vitamin E is somewhere in its presence.
Right.
And so that's another potential downside of PUFAs. Excessive PUFA consumption is it can deplete our vitamin E stores, because so much of the vitamin E is being used up throughout our body. Where, correct me if I'm wrong, it sounds like if there's like a battle, the vitamin E is like protecting the PUFA, but that the act of protecting the PUFA kind of gets rid of the vitamin E, but the PUFA is still there.
Yes.
Okay.
So I forget how many turnovers vitamin E can do. Like, vitamin E and vitamin C, they do it. They can do a number of turnovers to reduce something that's oxidized. So they then become oxidized, and then they have another molecule. Like for example, vitamin E can go to vitamin C, or to glutathione or whatever, and go through this whole cycle of antioxidant. But they can't just do that forever. Like eventually, they will get damaged and picked up somewhere and excreted.
Okay.
But the PUFA, it can get oxidized, and then say the vitamin E that like saved it gets carried away in a stretcher, to use your analogy. But that soldier is still on the battlefield until it's either burned in the mitochondria for energy or in some way gotten rid of.
Yeah.
And at any time, it can take another bullet.
Yeah.
And whether there's another medic on the field is,
Vitamin E. Yeah.
Right. So you're kind of like, and then people say, yeah, but if you're continuing to take in, for example, sunflower oil, you're getting a continual infusion of vitamin E. Yes, but it's always kind of one step behind. It's sort of like, you know how the post office sells like forever stamps?
Yeah.
And so they're getting revenue. And so their books look great, but they have a bunch of stamps out there for mail that they have to deliver,
Okay. Yeah.
and they're not going to get revenue to deliver that mail. So like if everybody stopped buying stamps, their revenue would stop, right? But they'd still have a backlog of stamps out there for mail they have to deliver. How are they going to pay for that? They can't. So it's like this sort of like a Ponzi scheme, but
Yeah.
like not like in a
Yeah.
malicious way or whatever.
Yeah.
So that's like with, it's just a question of how much mail did you promise to deliver, and how much like money do you have to do? It's like how many fatty acids that could be oxidized, and the conditions too. So you could have worse oxidizing conditions. If you consume the same amount of vitamin E, it'll get consumed faster,
Yeah.
and then you're in the same place.
Yeah. So I guess what would your best recommendation be? There's some talk in the bioenergetic community that like, okay, if you're going to go out to eat and consume something that's in a bunch of vegetable oil, it's good to take like vitamin E supplement with that to help reduce the probability of oxidation. What are your thoughts on that kind of advice?
Yeah, I don't really know how, like, so, you consume fat, it gets absorbed, it
Yeah.
gets exported by your, intestinal cells. Most of it will probably get taken up by the liver. Some of it will get taken up by other cells, and then it'll get re-exported and end up in either, blood triglycerides or blood cholesterol. And then eventually some of it will end up in, adipocyte storage.
Yeah.
So in that kind of first pass, like I just consumed this meal with fatty acids. Uh, yes, consuming vitamin E along with it is good, but again, like if that ends up in your fat storage, there could be some vitamin E with it in your fat stores, but it's not necessarily going to be the vitamin E. Like if you pop a pill or liquid or whatever vitamin E from a meal, that's not necessarily the same vitamin E that's going to end up, traveling at any given time with the oils you ate. So I would think of it more like a long-term thing. Estimate your intake of PUFA and kind of think about how much vitamin E. I mean, you can actually look at how much vitamin E is complexed with sunflower oil naturally. And, think of it like, okay, if my diet on, basically what I'm saying is I don't think it's a big deal. If you have one meal
Yeah. Yeah.
that has a lot of PUFA and you have a low PUFA diet, because I really think that long-term effects are more of what's going on than acute effects. Like you're not going to have any kind of health episode,
Yeah.
from the acute presence of a bolus of PUFA from a meal.
Right.
So you should really think of it like, am I consuming, often enough high PUFA foods that I should just supplement with vitamin E? That's the way I would think of it.
Yeah.
And maybe it's a good habit to just take vitamin E as a reminder when you eat a food like that, to kind of like keep in your head, like, oh, this is something to think about. Like
Yeah.
these kinds of foods have risks associated with them, but they're not really risks like that day or that week.
Right. Right. Okay. That's a good thinking, like more long-term repercussions of the storage of these PUFAs in your body.
Yeah. And the other thing is, when you said turnover, like 2 to 4 years, that's that's like a kind of like a chemical turnover, but you could like, if you could kind of tag every fatty acid you ever ate, right, throughout your whole life, there could be one in your body somewhere from like 10 years ago that just happened. You know what I mean? Like, because when you think of things like half-lives, the way things usually get consumed, whether it's drugs or nutrients or whatever, is like, oh, you lose half of it over this time, and then over that same amount of time you'll lose another half.
Yeah.
But every time it's a half of a smaller starting amount. So it's not a linear, the decrease slows down over time, and eventually you're losing half of a very small thing. And those things, then it starts to get really fuzzy.
Yeah.
Like if you take a drug, most of the drug is out of your system, say in 8 hours, But if you had a super sensitive instrument, you could detect some molecules of that drug days later.
Yeah.
But that might not be physiologically relevant.
Yeah.
But when it comes to stored fatty acids, so there could be, things from a really, really long time ago. Not that that's necessarily important, but just it's not it's not that your whole body just goes, okay, like a snake shedding its skin. Like
Yeah.
I'm getting rid of all of my fatty acids every 4 years and having brand new ones. That's not
Yeah.
the way to think about it.
Yeah. Okay. Good point. Okay. So one more point on the PUFAs, and it kind of relates back to your dissertation work. Um, One of the things that I have heard from other people is that PUFAs make fat cells insulin sensitive. And you think that that's a good thing. But then if you actually think about what that is implying, it's implying that PUFAs kind of shut off the fat signal's ability to know when it should stop growing in size. So it's able to stuff more energy inside that fat cell, and it can increase the chances of that fat cell expanding. And this was just one paper that was brought up about that. Um, and so I'm curious if that's kind of like a right way of thinking about this, because it would align with your dissertation findings where the rats that consumed the higher PUFA had a harder time with glucose disposal.
Mm-hmm. Yeah, there's there's a lot of different proposals for how it works, but it seems to be that, like, for example, one of them, that Ray talks about a lot, and that this is true, is that the normal competition called the Randall cycle
Yeah.
between fatty acids and glucose is not affected the same by all fatty acids. So if there are longer chain and more unsaturated fatty acids, they have more of a competitive effect with glucose because they require more of the transportation enzymes that are part of the signaling to the cell that, okay, we're going more with fatty acid use right now and less with sugar use than with shorter chain and more saturated fatty acids. So if that's and there's so many other potential ways that this could be happening, like when I was talking about some of the downstream metabolites of the PUFA, they can affect like insulin receptors and leptin receptors, because anything that is at all involved in inflammation or cell signaling usually affects those metabolic pathways, or it affects, like, say you have a receptor on the cell and that activates a protein inside a cell that, it's like whisper down the lane. Somewhere along that lane is something that is turned up or down by like prostaglandin E2 or whatever. So, but however the reason, it appears that if you were to just like infuse somebody's circulation, with PUFA, it will interfere with their glucose metabolism more than if you did it with like saturated fats. And again, this makes sense from the perspective of that's a signal to the body that there's dietary fat available. So like, okay, use less sugar, burn more fat, and also store more fat.
Yeah.
So, it makes the adipocytes, because it's like, okay, there's fat in our environment. So like, let's use it. Let's preserve the sugar for now. Because again, there would be times in like human history when there's not just a presence of carbohydrate all the time everywhere. So these things are all scarce at different times. So yeah, if you have a bunch of dietary fat, okay, like let's use a little bit less of the sugars that we have for now. Let's use more fat. And also, since fat is a kind of a slower storage, like let's store some of it for later because the fat's not always present.
Yeah. Okay. Okay, so that's kind of all I had on my notes about PUFA, but I'm curious if you had any more like recommendations to people. Like you said, you're not like hugely dogmatic on certain topics, but in general, how would you advise someone, like a friend who wanted to like improve their nutrition and lifestyle? What would your recommendations be in terms of like the fats that they consume?
Well, it's really easy because, um, it's actually extremely easy to just not cook with vegetable oil. And it's like that's such a low hanging fruit. And like if somebody, I don't know, eats a lot of salad or something and they're using like store salad dressing that's just sitting on the shelf, that's essentially just soybean oil with flavoring.
Right.
um that's it's pretty easy to make your own salad dressing, you know. That was something that I learned to do when I was doing like a raw vegan diet, because I went through a really big salad phase.
Yeah.
And I learned that, you know, if you have some sort of fat, like say olive oil or something, uh some type of acid, maybe a sweet thing like honey,
Yeah.
and a source of salt, it's like, okay, you have a good salad dressing. And then you could add like some other flavor, like an herb or, you know, whatever.
Yeah.
um so things like that are just super easy. I wouldn't really recommend people cut down. I mean, if you're consuming a ton of fatty fish because you think that's great and not just because you like it, then I
Yeah.
would be like, you know, just it's okay to eat fatty fish just once in a while. It's really not like, it's definitely not an everyday thing that you need to do.
Yeah.
Um, and I might even tell them like, well, you could replace that with shellfish because it's lower in fat.
Yeah.
And in terms of like minerals, you know, and other micronutrients, it has a better profile usually than fin fish. I always recommend organ meats. So I'm kind of, I'm kind of Paul Saladino adjacent with that one, like nose to tail. Um,
Yeah.
Eating.
Yeah.
Usually I just recommend liver because it's easy to cook.
Yeah.
Uh, there, I mean, tripe, you know, intestines and stuff are easy to cook too, but usually people get weirded out. I mean, tongue is not that weird. It's pretty, although that's a muscle, but it has a little bit of a different micronutrient profile than the skeletal muscle. Heart,
Yeah.
you know, because that has things like CoQ10. Um, so yeah, things like that. I mean, eating a diverse diet, because people don't eat a diverse diet now. You know, they just eat muscle meat. They get a lot of vegetable oils, and they don't get, you know, and you know, say like grass-fed animal fats or whatever, or you know,
Yeah.
people are throwing out egg yolks and doing things like that. I recommend against that. I don't think there's anything wrong with consuming egg yolks. Um, and yeah, that's pretty much it. And then it comes down to, because when I'm talking to somebody, when I was on strict diets, I would tell them like, oh, you know, don't eat this, don't eat that. It never really works anyway.
Yeah.
You kind of just have, I mean, unless there's somebody that is, and this would be extremely rare, but somebody who is interested in like a total diet manipulation and wants you to give them a total, but most people have things they like. So you just find things they like, and there's probably something that they can substitute, you know, if it's some product like, oh, I don't know, like if it's just a store product that, you know, the main, like I said, salad dressing or something where the main ingredient is vegetable oils. I would be like, oh, you know, why don't you find one where it's made with olive oil or something
Right.
like that?
Right. What are your thoughts on prioritizing like ruminant meat relative to like chicken and pork, which if you analyze like throughout the 1900s, we've transitioned the feed into largely corn and soy, and corn and soy are high in PUFA, and pigs and chickens are monogastric animals. So similar to us, the fat that they consume is kind of what is used throughout their body.
Mhm.
What are your thoughts there?
Yeah, I think beef is basically safer in this environment. I'm not sure. See, because poultry is, you know, quite a bit leaner in general than pork. So I don't know, like if you, because a lot of people, like say, you know, a bodybuilder or like a fitness person would consume like skinless, you know, trimmed chicken breast or something like that. So it's not really that much of an issue. But even on the micronutrient level, and I don't know about the amino acid profile. I don't know if it's all that different between the different meats. Uh, but yeah, beef is pretty much, or you know, ruminant meat in general. Most people, it's beef. You know, what if you have a source of bison or something? That's great. Um, yeah, that's in general better. And I wouldn't, if somebody's just eating pork all the time or like cooking bacon every morning, you know, it's probably not that good of an idea.
Yeah, yeah. I mean, I like pork and chicken, and so I include it regularly. And then when I do, I just make sure that it's pasture raised, so that way the animals have access to grasses and insects and bugs too. And again, it goes back to that like whole food form rather than just this concentrated in the pills. But okay, last question for you. Like you said, nose to tail. My sister and I are huge nose to tail advocates. I pretty much will eat anything, any organ. So what is Kyle's favorite off part, favorite organ from any animal? Anything is fair game.
Okay, so the organ itself, I would have to go with liver because I've just cooked it. So I'm very comfortable making it. I'm used to the flavors, so it doesn't seem off to me anymore. Whereas somebody that's more naive to eating liver, it's like can be a challenge.
Yeah.
So that's kind of a boring answer, but I'll give a more exciting answer,
Okay.
which is if you can find an authentic Chinese restaurant, you don't want to go there too often because they cook in vegetable oils, and the what they cook is very oily. So when you get it at the table, you can just see, you're like, wow, this is just vegetable oil. And it's usually they usually heat vegetable oils, like I think it's like sesame and maybe a couple other ones, and they infuse chilies into them, and it actually tastes, and it's very fragrant, super flavorful. And you can tell when it's an authentic Chinese place because they'll have things on the menu like sauteed pork kidney. Usually it's called in a dry wok, like
Yeah.
dry wok, and they'll have tripe. And so the spicy, and they'll have it sometimes with the Szechuan peppers, which are the kind that make your mouth numb. So that dish, sauteed pork kidney
Yeah.
in a dry wok with hot peppers, is my favorite organ dish.
That sounds really good. We actually had pork kidney the other week. I really like kidney. I again like all organs, but I'm sure it'd be so easy to make that with just butter or coconut oil or something like that. I don't know.
Yeah, it's a shame, because with the Chinese place, it's so integral to the cuisine to use the oils they use. The only thing you would lose some of that fragrant, like
Yeah.
aromatic quality of the sesame oil. But what you could do, I mean, they could probably figure out a way if they wanted to, which they're obviously not going to cater to like some white guy like me. But that, you could probably approximate it pretty closely if you really understood the dish and you used like coconut oil as the base and then just a little bit of sesame
Yeah.
seed oil for fragrance.
Oh yeah, absolutely. Yeah, to just give that little, yeah. Okay. Well, cool. Thank you, Kyle, for hopping on today. We'll have to get you back on to talk about some other topics at some point.
Yeah.
But again, appreciate it and hope you have a great rest of your evening. Thank you.
Yeah. Thank you.